So for this part of the lectures on advanced experimental techniques, we're not going to talk so much about doing experiments, but just give you some hints and tips for actually presenting your data. So in the module, you're going to have to go and give several presentations, one for each little experiment you go and do, as well as go and write your formal report. And that, of course, will include data that you present. So I've spotted several errors over the years that are kind of common to lots of students. So I just want to try and clear up some of the things about how, good ways of actually presenting your data. So let's take a look at some data. So this really ought to be things you already know about, but I still see these sorts of mistakes being made on a fairly regular basis. So you need to, when you're looking at this graph, also imagine that you're looking at this in a lecture theatre rather than on your laptop screen. So the question is, what's wrong with this graph? What are all the possible mistakes that have been made here? So let's start going through some of the problems. So the first thing, are those axes labels really easy to understand and read? So if you've got a large number of leading zeros, it's really quite tricky to work out quickly what the order of magnitude of the number you're looking at is. Likewise, your axis labels should actually be labeled as something meaningful. A and B does not really help you very much, and it doesn't give you the unit of the measurements in, or what it means. Then you might wonder, well, what's all this white space doing here? Why are we showing these axes in the way they are with a huge gap of the data? Is that meaningful? One of my personal bugbears is the size of the font. If you were seeing this in a lecture theatre, you would not be able to read these labels from the back. They would simply just be too small. Then another common gotcha is the title on the graph. Well, what does that mean? And more to the point, is it really suggesting that you've got a systematic approach to how you're numbering your samples? And does it actually convey any useful information at all to the reader of this presentation? How would they know what batch you're talking about here? Good run, so presumably there were some bad runs. Try two, so maybe you had more than one shot at trying to do this measurement. Who knows? And then you notice that the dots are all joined together with a line. And the question is, well, what does that line actually mean? When you put a line on a graph, you're implying that you know that the result of the measurement is the same at all the points along that line, which if you haven't actually measured it is a little bit questionable, to put it mildly. So does that mean you've actually got a continuous measurement and you've just highlighted some particular points in that continuous measurement? Almost certainly not. Is it a guide to the eye? Well, how would the person reading the slide know that? And then you also spot there is one kind of point which seems to be off the trend of the rest of them. And the question is, well, what happened there? It's being treated the same as all the other data points. So is it real? Is that something you're actually looking at or looking for? Or is it actually more likely to be an artifact? So lots of things that could be wrong. So here's the same data, but now with a slightly more sensible set of way of presenting it. So first of all, we can see that the axes labels are now numbers that are a reasonable size. So generally speaking, what you want to do is make the size of the numbers on your axis label somewhere between 0.1 and 1,000, and then use the appropriate SI prefixes on your units in order to go and get it into that right size. So you can see, well, an axis label here that tells you what it's measuring. It's also got some units. And you see I've used micro ohm meters so that I can make my numbers on the y-axis actually be sensible. And on the bottom axis again, we've got it in reasonable units with a descriptive label and the SI unit for that label. We've got a better presentation. We've adjusted the axis limits in order to show the data clearly. We've labeled a linear fit through it, so we've actually put a proper fit to it, not just joined the dots and said what that is. And our legend actually says exactly what the measurement is of, and the title to the plot, give some information about what the plot might be about, at least a measurement date and some idea of what the measurement is. And then finally, we've identified the outlier point and indicated what might be wrong for it. You'll also see that all the fonts here are now big enough that there's some chance you could actually read it from the back of a room. If what you're actually interested in is looking at a deviation from a fit or from a trend, then you might want to think about taking off the trend line from the data. So that's what we've done here. So now that's still the same linear fit, but it's set so that we're comparing relative to that fit. So the fit now runs along the y=0 line, and the data is scattered about it apart from that one outlier point. So again, thinking a bit about what it is you're trying to make the point. What's the story you're trying to tell with this graph? Therefore determines how you want to go and present it. So various things about producing sensible graphs. So first of all, set the point size to be the same as the final size, or set the plot size to be the same as the final size. So if you're making a plot to go in a report, then and you know that you're doing to column width, and therefore your column width is about 8.5 centimeters. Set the plot size in Origin or whatever it is you're using to be 8.5 centimeters or possibly 17 centimeters wide, and then you can go and scale everything else so it looks proportionate for that size. Watch out what the minimum point size you're actually using, and try and keep it the same as the surrounding text, and also just trying to make sure that you use a similar font to the surrounding font as well. It just looks nicer. So in particular, I'd say never go below 18-point fonts on a PowerPoint presentation, and in text for report, 11 points is a sensible minimum font size. It's very easy when you're making a figure to set the plot size to be a whole A4 page and then sort of set the fonts to be 10 or 12 points. And it looks absolutely fine on your computer screen. But then when you shrink the whole thing down to a half column, to a one column figure, then you just suddenly realize that font size is now about 4 points and completely illegible. And if you find you're having to zoom into the screen in order to read the graph, that's a definite sign that you've got the fonts too small. So things to check. Check you've got a title to the figure, it's properly descriptive. Check the axes labels are actually a physical quantity in units, and you're using SI prefixes to avoid having too many zeros. So as I said, try and keep the numbers always between 0.1 and 1,000. That's much easier to actually go and work out what those numbers actually mean, and then label the units accordingly. Colors. Again, this is a common one where you're switching between working on a computer screen and then projecting it. So most projectors are much worse at giving high saturation to colors. So the colors will end up looking quite washed out. And what that means is you really want to avoid having light colors on white backgrounds, and likewise avoid having dark colors on dark backgrounds. And then also think about people who might be colorblind. So avoid using red and green with a similar tone, because the difference between those lines is going to be almost impossible to distinguish for someone who's colorblind. And red-green colorblindness is the most common form of colorblindness. So it's the one you definitely want to watch. There are a few other ones, but they're significantly less common. So it's all about making sure you've got the similar, about watching for tone. If you're working for something which is going to be printed out on paper, then another thing to think about is whether, if it's printed out on a grayscale, is it going to end up all looking the same? So try to get a color range which is going to come out to be different when it's converted to grey if you're doing it for paper. Again, lines indicate a continuous variable, so a fit to a theory. If it's just supposed to be a guide to the eye, then say so in the caption. And try as far as possible to avoid doing join the dots. Join the dots is nearly never the right way of presenting your data. The only point at which you might want to go and do is if you've got lots of different data sets on the screen at the same time, where you definitely need some way of showing which sets of points join together. But again, in general, if you're just putting large amounts of raw data, think, why are you actually doing that? Is that really a good way of showing what's changing as you change the as you look at different measurements? Perhaps there's some parameter you can measure off off the data you've collected that is going to be more revealing about how the data changes with the measurement you're doing. Error bars. If you know the error, then plot it, unless it's smaller than the point marker. And hopefully most of the time you do know what the error bar should be for your data. So it's always a good idea to plot the error bars if you actually can know what the error actually is. So another important point to doing good graphs, and this applies more to formal reports than it does to presentations. So in presentations, we don't normally provide a detailed caption. The other thing I should say with presentations is it's not normal to number your figures. So occasionally I sort of see going through a presentation, someone's got sort of figure 3, figure 4, figure 5 on their graphs. We don't normally do that. Normally what you do is just simply have 1 or 2 graphs per slide, and you don't have a caption in the same way you do with a written on paper sort of graph. You maybe have a little bit of explanation text beside it. But if you are doing a written report, then all your figures should be captions. And one of the really important things about writing scientific work is you should be thinking about how the reader is going to actually read this. So a good piece of advice is to think that you're going to expect your reader to read your title, your abstract, then all the figure captions and look at the figures, and then your conclusions. So that means figure captions should be as standalone as far as possible. So you should get the general idea of what's going on from reading, as I said, the abstract, the figure captions, and the conclusion. So this is one of the really common things I see that changes between typically BSc project reports and MPhys project reports. Quite often with the BSc project report, the captions end up being a one-line description. So for example, the plots we were just looking at, you have a caption which reads graph of resistance versus temperature. Well, assuming that you've labeled your axes correctly, I can work out that it's resistance versus temperature because I can read the axis labels. And I kind of know it's a graph because I can see that it's a graph. So that figure caption is really not adding anything at all useful to my understanding of your figure. On the other hand, a figure caption that says what the interpretation of the measurement is, so in this case, temperature dependence of resistivity for a particular sample, prepared in a certain way, indicating a little bit of a conclusion, and the conditions under which it was measurement, and only then do I refer maybe perhaps to some even more detailed discussion in the text. So getting your figure captions so they're useful is a really important skill. The other thing I should want to say about both presentations and about writing formal reports is that you really want to think about what the story is that you're telling here. So what is the message that you want the reader to take away at the end of this presentation or at the end of the report you've written? What are they going to know about? What are you going to prove to them? And then what you want to think is, okay, if I want to go and prove this fact to them, or I want to show them this particular conclusion, what evidence do I need to provide them with? And everything else in the content of your report or your presentation should be building up the evidence that you require in order to go and make the point you're trying to make. That means that you shouldn't just be showing data because you measured it. For every figure in every plot you make, you want to say, well, why am I showing this data? And how does it support the conclusion I'm trying to come to? And the other thing you want to think about is how do you always want to like storyboard out your work. What's the story here? Where's the reader start from? How do you take them on the journey of understanding your conclusion? That's not the same thing as saying do it in the same order that you actually did the measurements in. Often we find that we do experiments and we collect the data at the end of it, and then and only then do we get to the point of being able to understand how it all hangs together. And so the order in which you present data is not necessarily the order in which you took it, but it is making the point you're trying to, or the conclusion you're trying to come to. So that's the thing you actually have to think about, is the actual structure of the document you're writing or the presentation you're giving. Always be thinking about helping the reader or the listener to understand where you're going with this. So, for example, in a presentation, a common pattern would be to go and say, well, I'm going to present you my work on this. I'm going to tell you these 3 things. And then as you go through it, you say, so the first thing I'm telling you about is this. And now the second thing I'm telling you about is that. And then the third thing I'm telling you about is this other thing. And then to conclude with, in this talk, I've gone and talked about these 3 things. And this is the 3 conclusions we come to as a result of this. And the reason I'm saying 3 is there is this thing called the rule of 3 that it just makes a good flow and a good structure to a presentation. And it seems that the human brain is well adapted to holding on to 3 facts at a time. So if you structure your thing in groups of 3, it kind of flows very nicely. And if you listen to a good political speech, for example, you see that that's what they're doing. Everything goes in groups of 3. Points are made in groups of 3. And that's because it's how your typical human brain can process information. And the same is true when you're presenting your work. So really be thinking about the structure as you're trying to write a report or you're trying to give a presentation. In particular, presentations should always obviously have a title. If the presentation is going to be more than about 10 minutes, you almost certainly want to have a contents slide or an overview slide. For very short presentations, it's a bit more optional. But if you've got a series of sections, you might want to think about how to do that. If you don't have an overview slide, you can sort of integrate it into your actual slides by having some kind of obvious progression. So one technique I've used in the past is I put the contents in a column down the side of my slide, and I just highlight which part of the contents I'm on as I go through it. So the reader can always see how far through this story I am and how it relates what I'm saying on this slide to the rest of my story. And then at the end of your presentation, you should always go and have a conclusion slide. What's the take-home message? And again, something that often is done in research talks is with each conclusion, you give a little postage stamp snapshot of the key graph or the key graphic you used for that particular point during the presentation. And that helps the reader key back to understanding the relationship between that conclusion and the key point that they made. Okay, so error analysis is another area which often causes some disquiet or concern as to how to go and do it. So the standard thing, 1.7329 plus or minus 0.425 types of physics students in the world. Those that quote the results to improbable levels of precision, and those that pass this module. Okay, I cannot stress this enough. By now, you really ought to know that uncertainties can only ever be quoted to one significant figure, unless you really have huge amounts of stats. If you go and look in the textbooks, then it'll tell you that in order to quote an uncertainty to 2 decimal places, so to 2 significant figures, so in other words, to have the uncertainty in the uncertainty to 1%, you need something like 5,001 separate measurements. And it's very unlikely that anything you're going to do is going to have 5,001 separate measurements, unless you've really gone and done your stats carefully. So unless you know better, you always quote the uncertainty to 1 significant figure. And then there is no point quoting an answer to more decimal places than the uncertainty, because you've got no way of knowing what the decimal places after that 1 digit of uncertainty actually is. So you should always be quoting your answers the same number of decimal places as the error you've quoted in your answer. And just because the computer sticks out that number, then doesn't mean the computer's right, doesn't mean it's being sensible. So if you quote a number, then really you should have an uncertainty as well. You should always be thinking, every time you've come to a conclusion with a number, you should be thinking, okay, what's the uncertainty in that? What confidence? And it may be that you can't do an accurate error analysis and calculation, but you should still be thinking, well, what's a reasonable level of uncertainty there, based on your understanding of how you've calculated that number. And again, just to reiterate, just because the computer returns answers to 8 decimal places does not make it right. Computers are intrinsically stupid. So if you're fitting a model to some data set, then you're going to have some fitting parameters. They really ought to have uncertainties, so you should quote them. So wherever you can get an uncertainty out of a fit, then you should really go and do that. It's really easy to get too paranoid over your error analysis, however, and kind of go over the top with it. In most cases, you can apply some common sense to what's going on. So you can ask some questions. Are there some quantities where the uncertainties are small compared to the other ones? You can just simply ignore them because they don't make a big enough difference. Is there some quantity that completely dominates over your calculation? In that case, usually the error also dominates. So if you've got an equation with a bunch of things which are not raised to any particular power, and then something which is raised to the 5th power, probably the uncertainty in the thing raised to the 5th power is going to be the thing that's going to control the uncertainty overall. Because obviously, if you just do the maths on that, then the uncertainty in that is going to be multiplied by a factor of 5. So the chances are that it's going to start winning out over the other quantities. A good way of dealing with, to get a good combined error calculation, you can sort of simply do numerically. You can simply do by substituting in the uncertainty in each quantity into your equation. So for example, if you have an equation z equals a plus b, you do, and you want to know what the delta z you should use is, you can say, well, let's do a plus delta a plus b, and let's do a plus b plus delta b, and then pick whichever one gives you the biggest difference from just a plus b. So whichever of the delta a or the delta b is giving you the bigger answer. Now, of course, here it's trivial. You could just look at it and go, well, it's either a or it's b. But if the equation is quite complicated, that may not be so obvious. So you can just run it through and numerically estimate what one's going to win out. And then use that. That's a perfectly valid way of estimating the uncertainty, because remember, you're probably at best, very, very best, you're probably only estimating your uncertainty to 1 to 10%, because you're only quoting it to one significant figure. So really on this scale of things, does it matter? The other thing you want to be really careful about is systematic errors. So when you're doing your measurement, it might seem kind of an obvious way to go and do things is to do it in a systematic order. So say you had to go and measure a sample at a range of different temperatures. It might seem like the really obvious thing to go and do is start at the lowest temperature and work your way up, going to bigger and bigger temperatures. The problem is you then have the possibility of introducing a systematic error, because what you don't know is whether any change you're seeing is due to a change in the temperature you're measuring, or simply due to the fact that it's later in the day because you spent all your time doing the previous measurements. So instead of just working systematically through a data set, randomize the order in which you go and do the measurements, unless there's a good reason not to. So what happens then, if there is a systematic error due to the time of day, by randomizing the order of the thing you're controlling, the temperature, or I don't know, the sample thickness or the sample number, whatever it is, by randomizing that, and then when you analyze the data in terms of a change over the sample temperature, thickness, sample number, whatever else, the order in which you're doing is then automatically randomized, and therefore your systematic error, because you did things one after the other, and it was varying over the time of the day, ends up becoming a random error, and then you see that as a scatter in your measurement, and it'll be picked up when you try fitting data if you have large uncertainties. Of course, it'll give you large uncertainties in your answers, but at least you can see that there's a problem with it, and so you understand that you have a some kind of larger uncertainty than you were expecting, and then you're in a position to be able to address it. Whereas if you don't do that, you can easily miss the systematic error and silently get the wrong answer. And that's almost worse than getting the right answer but with a large uncertainty. So again, that's a little bit about thinking about how you do the experiment. Okay, so some final words on good practice. So read the recommended course text, which is this book by Squires. I don't know if it's available online, Normally, I'd be saying go get it out of the undergraduate teaching lab, but of course that's not an option this year. But if you can get hold of a copy, that's one of the recommended texts. So you want to be thinking about what you're doing as you do it. So don't just go into the mode of collect data and don't stop and think about what that data means. Again, this is things we've taught you in first and second year lab. So that's obviously doing things like a pilot experiment, but it means carrying on thinking as you carry on doing the measurement, and try and work out what's going on. Record everything. There's never too much to record. So every setting on every instrument you use, also write down the good ideas, the bad ideas. So if when you're keeping a lab book, you think you're doing this to write it up neatly. So the absolute killer is I find students working things out on paper and then they say, oh yes, but I'm going to write it up neatly in my lab book. Your lab book is not supposed to be neat. Your lab book is a working document that has working all your ideas as you go through. Now, obviously it helps if it's not a complete disorganized mess, but a good lab book I expect to see is something where I can, well, firstly work out what you were doing on a given day, but also what you were trying to do, what you thought you were doing, what you thought was going on, and whether you thought it was actually doing what you expected it to go and do. And so I expect to see a certain amount of crossings out. I expect to see a certain amount of noting down questions to yourself. Lab books are not supposed to be neat and tidy. And again, it's to distinguish very carefully between a lab book and a formal report. So a lab book, I don't really expect to sort of see a nice thing that has introduction and method, and then data, and then discussion, and then conclusion. Because very rarely do you do an experiment like that. So yes, I mean, at the start of an experiment, I'd like to see in a lab book some notes about what the background physics. So typically, the supervisor of each experiment will give you some background information about the physics of the experiment, the purpose of what you're trying to do, maybe about how it works, and I expect to see some evidence that you've been taking notes when you met with your supervisor for the first time. Then, yes, you might have a sketch of or a schematic of the equipment, marking the point at which you first used it. And then we expect to start seeing a mix of data and interim conclusions, and we look for evidence of interim conclusions. And then at various points, you'll probably want to go and stop and note down what you think the conclusions are. But you don't have this nice division into these regular sections, and things are, as I say, I don't expect it to be written out super neatly. The reason why you want to record everything, however, comes down to the point that you might well get to the point after you've finished doing your measurements, and then you start realizing that the thing's not fitting, or the analysis isn't going quite as you expect it to go and go. And then you might realize that in fact this is because you changed some parameter in your experiment that at the time you didn't think was important. If you've made a note of every single parameter you've got access to, then you're in a position to be able to go and say, well, hang on, look, there's correlation here. And that could even be as simple as just noting down what time of day you were doing a particular measurement on. So the more information you can record about how you're doing your measurements, how you're doing the analysis, the more likely you are to pick up artifacts in what you're doing. And of course, sometimes you have completely crazy ideas, but record them because you never know what it might get you. So this is Andre Geim, who is relatively famous in the physics community for winning the Nobel Prize in Physics for the discovery of graphene. But he also has the Ig Nobel Prize in Physics because he decided to go, when he was working at the Nijmegen Institute for High Magnetic Fields, to have a shot at levitating a frog. So this is using the diamagnetic response of a frog tissue to a large magnetic field and seeing whether it's enough to go and cause the frog to levitate. So I think, as far as I know, he's the only person to ever win both the Nobel Prize and the Ig Nobel Prize in the same subject. So there you go, a bit of fame.